Hello students. So today we discuss about the two parts of photosynthesis. Two reaction make up photosynthesis. So first is light reaction. It is also known as light dependent reaction. So produces energy from solar power means photon in the form of ATP and NADPH. So second is dark reaction. It's also known as Kelvin cycle. Light reaction, independent reaction. It is also known as the independent reaction. And another name is carbon fixation or C3 fixation. Use energy ATP NADPH from light reaction to make glucose or sugar. So first we discuss about light reaction. Light reaction here photophosphorylation, formation of ATP in the chloroplast in presence of light is known as photophosphorylation, and photosynthesis is divided into two types: light dependent reaction, then light reaction or photophosphorylation. And another part of photosynthesis is dark reaction means Kelvin cycle. It is known as light independent reaction or C3 pathway. So here first point is photophosphorylation take place into two pathway means light reaction is here divided into two pathway, cyclic and non-cyclic. So this light reaction occur in thylakoid membrane. During the light reaction there are two possible routes for electron flow. First route is cyclic electron flow and another is non-cyclic electron flow. Cyclic electron flow. In a cyclic electron flow, first occur in thylakoid membrane. Use photosystem first only. Photosystem first only. Take place in bacteria. Then photosystem 1 means P700 nm wavelength means reaction center chlorophyll A. Use electron transfer chain ETC. Generate ATP only. Only ATP generate from this cyclic electronic flow. Means here adenosine diphosphate plus inorganic phosphate and then they convert it into adenosine triphosphate. Okay. So first cyclic form. Here cyclic form means photophosphorylation take place. So first way, that way is known as the cyclic. So first only system, pigment system 1 is only here. The reaction center is made by P700 Mn chlorophyll. Then the light falls on photosystem 1. So here sun and light or photons falls on photosystem 1 and it get, okay, so photosystem 1 get excited the as well as the accessory pigment pass on its energy to reaction center P700 which enters energy and energy rich electrons there. So these electrons are accepted by electron acceptor. It is known as primary electron acceptor, and primary electron acceptor is FRS. The meaning of EFR is pyridoxine reducting substance. Then from EFRs, electron is transmitted through EFD, and EFD means pyridoxine to cytochrome B, cytochrome B to cytochrome B6, from cytochrome B6 to cytochrome F, and cytochrome F to plastocyanin means PC, and from plastocyanin the electron returns back to P700 chlorophyll A molecule. So during electron transfer between cytochrome B6 and cytochrome F. So here important point is here, during electron transfer between cytochrome B6 and cytochrome F, the energy is released. The energy is released in the form of ATP. So here process of synthesis of ATP from ADP in presence of light during cyclic transfer of electron. Why it is known as cyclic electron flow? Because initial donor and final acceptor, initial donor and final electron acceptor is same, and that is photosystem 1 chlorophyll A, P700 Mn is a common, means initial donor as well as final acceptor. Hence it is known as cyclic electronic flow. Non-cyclic electron flow. ADP plus inorganic phosphate, then they convert it into adenosine triphosphate. NADPH plus H, NADPH. Oxygen comes from splitting of water, not from carbon dioxide. So here non-cyclic electron flow is also known as Z scheme, occurs under aerobic condition like high carbon dioxide concentration. Process of synthesis of ATP and NADPH2 from ADP and NADP in presence of light during non-cyclic transfer of electrons. So location of this cycle in grana in thylakoid membrane. Photolysis of water in non-cyclic photophosphorylation. First important point is here, photolysis of water is occur. ATP and NADPH2 synthesize, means end products are ATP and NADPH2, but absent in bacteria. And non-cyclic photophosphorylation, they involves the steps photo excitation of photosystem second, means P680 nm, flow of electrons from photosystem second to photosystem first through electron transfer system, synthesis of ATP, then photo excitation of PS1, P700, and last is photolysis of water. So all these are the introduction about the non-cyclic electron flow. It involves both photosystem 1 and photosystem 2. Electron transfer system start with the release of electrons from photosystem second. Photo excitation of photosystem second absorb light and get excited, which results in ionization of chlorophyll A, and a high energy electron is transferred from PS second. Energy rich electron is accepted by primary acceptor, and primary acceptor is known as coenzyme Q-non. It is also known as PQ or COQ. Electrons from coenzyme Q-non move down through various electron carrier and release energy from coenzyme Q-non. Electron transfer to plastoquinone mean PQ. PQ from plastoquinone electron transfer to cytochrome complex mean cytochrome B and cytochrome F. From cytochrome complex electron transfer to PC plastocyanin. From plastocyanin or from PC, the electrons are finally accepted by ionized chlorophyll A means PS1. Okay, so here PS1 photo oxidation or photo excitation of PS1. It get excited, photosystem 1 excited and transfer electron reach first acceptor FRS. Electron from FRS electron are transmitted to FD, and FD means pyridoxine. Reducing pyridoxine transfer electron to NADP, and the protons are accepted by NADP to NADPH2 with the help of enzyme, and the name of enzyme is NADP reductase enzyme. Okay, so here photolysis of water. Photolysis of water is occur because splitting of water break down into H plus, OH minus and release oxygen. So location of complex for photolysis of water, location or site is inner membrane of thylakoid, and NADPH reducing enzyme is located on outer membrane means thylakoid is made up from lipid, out membrane of thylakoid. So here actually not a two membrane, bilayered lipid membrane. So outer lipid, on a outer lipid membrane the NAD reductase enzyme is located, and splitting hydrolysis of water, splitting water or photolysis of water take place inner lipid membrane of thylakoid means water splitting of water take place in lumen of thylakoid. Then here, so your sun's trap, then primary electron acceptor receive, then all this electron transfer via the electron transfer chain to P700, primary electron acceptor FRD receive this electron and then transfer into the NADPH2, and splitting of water recover the lost electron in P680. Then non-cyclic electron flow occurs in thylakoid membrane. Use photosystem first and second, means both photosystems are used in non-cyclic electron flow. Then P680 reaction center in PS second, means photosystem second, chlorophyll A, and P700 reaction center present in photosystem first. Use electron transfer chain is known as the ETC. Generate oxygen and ATP and NADPH2. So end product of non-cyclic electron flow is they release, because photolysis of water take place, so they release oxygen, and the end products are ATP, generate ATP and NADPH. So here comparison of cyclic and non-cyclic pathway. So first difference. So first, cyclic take place in all photoautotrophs, but non-cyclic does not take place in bacteria. In case of bacteria, non-cyclic photophosphorylation absent, but in cyclic take place in all photoautotrophs. Then in cyclic, initial donor and final acceptor is same. Final acceptor and initial donor is same in cyclic, but in a non-cyclic, initial donor and final acceptor is different. In case of cyclic, they involves only one photosystem, that is photosystem first, but in a non-cyclic, they involve both photosystem first and photosystem second. In cyclic, light energy is absorbed is at 700 nm wavelength, but in a non-cyclic, light energy is absorbed at 680 to 700 nm wavelength. Then in cyclic, energy in the form of ATP, so products in cyclic photophosphorylation, but in a non-cyclic way, non-cyclic photophosphorylation, the products like ATP, NADPH2 and water. In cyclic, no photolysis of water take place, but in a non-cyclic, photolysis of water take place. So photolysis of water take place in first acceptor is FRS in cyclic, but in a non-cyclic, first acceptor is COQ, coenzyme Q9. Then in a cyclic, only one electron is released, but in non-cyclic, 4 electrons are released. Then in a cyclic transfer of electrons, but in a non-cyclic transfer of electron, hence it is known as Z scheme. It is another name, another way is known as the Z scheme. So here is the difference between cyclic and non-cyclic photophosphorylation. So important points are here: pathway of electron, cyclic forming cyclic, non-cyclic form in non-cyclic. Then first electron donor, in case of the cyclic, photosystem first. Okay, photosystem first donate electron to the primary electron acceptor. But in a non-cyclic, donor, first electron donor, water. Okay, but in last electron acceptor in cyclic, photosystem 1. And in case of the non-cyclic, NADP, because NADP bind with the H plus and converted into the NADPH2 with the influence of the enzyme NADP reductase enzyme. So here last acceptor is NADP in non-cyclic, and in a cyclic, photosystem 1 is last electron acceptor. Then end product of cyclic and non-cyclic. In a cyclic, only generate ATP, but in a non-cyclic, ATP as well as NADPH2 and water, what after the splitting of water, oxygen is the product in non-cyclic. In non-cyclic, ATP, NADPH2 and oxygen. Then number of photosystems present or involved in cyclic photophosphorylation, photosystem 1 only, but in a non-cyclic, photosystem 1 and photosystem 2.